Hi, we're a group of 6 early career researchers in anxiety who have come together to write a review on the current state of play for anxiety genetics. One of our numbers, Isham, isn't available for this recording, but the other 5 of us are going to take you through the main points. So anxiety is characterized by feelings of unease, tension, and worry, alongside physiological arousal and anticipation of threat or ambiguity. Anxiety is interesting in that it's a universally experienced thing, and it can even be adaptive. But when it's experienced disproportionately, intensely, or for a prolonged period of time, it can be really harmful or maladaptive. This is particularly troubling because anxiety disorders are amongst the most common class of mental health disorders, with a global lifetime prevalence of 16%. They have a particularly early age of onset and a chronic course across the lifetime. In addition to dimensional experiences of anxiety, the DSM-5 describes 7 anxiety disorders. 2 of these, selective mutism and separation anxiety, are specific to childhood. The remaining 5, generalized anxiety disorder, panic disorder, specific phobia, social phobia, and agoraphobia, can be experienced throughout the lifetime. All of these, with the exception of social phobias, are more commonly experienced in women than men. Now, we know that anxiety is complex. It's influenced by a combination of biological and environmental factors, with genetic influences being amongst the best substantiated risk factors. The leading etiologic model for anxiety to date is the diathesis stress hypothesis. This suggests that genes and environmental stresses, both independently and in combination, increase an individual's liability to developing disorders. Now, environmental factors that have been associated with risk for anxiety include low socioeconomic status, parental conflict, and childhood maltreatment. It's really important to remember that environmental factors are themselves somewhat heritable, and genetic expression might be shaped by the environment and by life experiences via gene-environment interplay. Thus, associations between environmental factors and anxiety might reflect effects of genetic and environmental confounding rather than underlying causal relationships.
As Kirstin just mentioned, anxiety is the most common mental illness, even having been treated as a discrete diagnosis only relatively recently in the DSM-3. The volume of these diagnoses contributes to the cost of healthcare systems, which is further exacerbated by very high persistence across the lifespan, as well as an early onset of diagnosis. Together with depression, which frequently presents comorbid with anxiety, anxiety disorders cost an estimated $90 billion in personal healthcare costs within the United States alone. But there's also substantial costs to human health. Anxiety disorders present frequently comorbid with depression, as I just mentioned. It's occurs about 50% of the time in anxiety disorders. There's a depression diagnosis as well. There's substantial overlap also with stress-related disorders, such as post-traumatic stress disorder, which is formally considered an anxiety disorder until the DSM-4, and obsessive compulsive disorders as well. Substance use disorders are also often present comorbid with anxiety diagnosis. They also often present an overlap with somatic conditions such as epilepsy and irritable bowel syndrome. And there can also be a substantial impact on mortality. You'll often see a comorbid diagnosis with heart disease and with cancer. And when these co-occur, particularly also when anxiety, depression co-occur with a cancer diagnosis or a heart disease, you'll see a poor outcomes in disease. And it can be complicated to diagnose anxiety disorder in someone suffering from a cancer diagnosis or a heart disease, as someone with a life-threatening condition might have a normative response with anxiety or depression. There are treatments available in the form of psychotherapeutics, pharmacotherapies, and there's cognitive behavioral therapy has shown some effectiveness in anxiety disorders, but there are also some concerns about treatment responses. Only half of adults and 1/3 of children will respond to existing treatments, and few ever remain in remission. There's also many with anxiety disorders that don't receive treatment. Untreated anxiety disorders have particularly bad outcomes with impaired general functioning, social relationships, and employment that can last for decades. Improving both access and treatment efficacy is essential for anxiety disorders.
Twin studies provide an elegant natural experiment to investigate the relative influences of nature and nurture on anxiety disorders. By comparing identical and non-identical twins, we can disentangle the proportion of variation in anxiety disorders that's due to genetics, shared environment, and non-shared environment. Shared environmental influences are those that increase the resemblance among family members, whereas non-shared environmental influences are individual specific and those make family members different from one another. For anxiety disorders, genetic influences are moderate. Heritability estimates range between 20 to 60%. Environmental influences are primarily non-shared, although shared environmental effects are often detected in child anxiety. So in addition to this core finding of the heritability of anxiety, twin studies have shed light on at least 3 other fascinating topics related to anxiety. First of all, they've shed light on the genetics underlying the comorbidities that anxiety shares. So the genetic correlation with depression is nearly 100%, which suggests that anxiety and depression are different manifestations of very similar genetic risk. Secondly, twin studies have informed us about how anxiety manifests across the life course and why. In fact, anxiety disorders tend to have their onset in childhood, and twin studies show that continuity in anxiety from childhood through to adulthood is predominantly driven by a core stable set of genetic factors. Thirdly, twin studies have revealed interesting examples of interplay between genetic risk for anxiety and the social environment. For example, a well-replicated finding is that genes involved in anxiety in young people tend to evoke elevated levels of maternal control. Genomic tools provide new, exciting opportunities to extend twin study findings, including about gene-environment interplay.
The heritability of anxiety disorders is mainly based on a large number of genetic variations with only small effect sizes. Such variations are often not restricted to a specific anxiety disorder subtype. They more often influence a variety of different anxiety disorders, also explaining the high comorbidity rates between them. Aiming to uncover the complex genetic background of pathological anxiety, several genome-wide association studies have been performed on different anxiety disorders. However, due to small sample sizes and the high genetic heterogeneity of diagnosed anxiety phenotypes, the majority of results have been negative or not reproducible. To overcome diagnostic boundaries and sample size limitations, recent genome-wide association studies started analyzing the different anxiety disorder subtypes together or used dimensional anxiety symptoms. In doing so, 4 large and well-powered genome-wide association studies has been performed from the ANGST Consortium, the Danish National Register Study IPSYCH, the 1 Million Veteran Program, and the UK Biobank, reaching sample sizes ranging from 18,000 up to more than 190,000 individuals. Together, these studies lead to the identification of 15 genome-wide significant associated loci. Of those, 12 mapped to different gene regions. However, to further uncover the genetic makeup of anxiety disorders, more genome-wide association studies in large, well-powered sample sizes and on well-defined anxiety phenotypes are necessary.
The first GWAS from the Anxiety Workgroup in PGC is currently underway and will be presented at this conference. In the future, large-scale consortia collaborations and methodological developments in molecular genetics hold great promise for the future of anxiety genetics research. Guided by findings from quantitative genetics, we expect novel findings on the genetics across the lifespan and on possible gene-environment interplay. Core features of anxiety disorders will likely take the field of anxiety genetics in a different direction from that of other psychiatric disorders. The early age of onset and high chronicity highlight the need for studies among young people and across development. Additionally, the phenotypic complexity of anxiety and its subtypes and the high level of shared etiology with other disorders and traits speak to a need for genetic investigations on underlying or intermediate phenotypes as well as on related phenotypes. The results of anxiety GWAS open the door for a range of new possibilities for research. One flexible approach is the use of polygenic scores. The richness of longitudinal cohort studies like ELSPAC and MOBA provide a unique possibility to map how genetic risk manifests across development, to study the gene-environment interplay, and to estimate indirect genetic effects across generations. Genetic loci and polygenic scores can also be used to unravel causal relationships using Mendelian randomization. Genetic variation predates any behavioral or even neural variation. Understanding the underlying genetic architecture of anxiety and associated phenotypes will pave the way to better understanding of the complex downstream relationship between genes, brain, behaviors, and environments. The field of anxiety genetics is at a pivotal point of this understanding. We hope that future work will build on existing research to bridge the gap between disorders, prevention, and treatment. Thank you so much for listening.